Normally they're castrated when they're 50 days old. And
So
at
we
that
have a
point,
few still here
she
from
had
the
no
original.
idea that
Okay, so I don't know if we told you this yet. How did we get these animals so healthy?
Yeah, that's what I was wondering there.
Right. It was not an easy undertaking. Anyway, so what we did was we found a single source of sheep and bought 135 ewes and 2 rams and started breeding and had to figure out a process where we needed to know exactly when they were due. So we would breed the ewes only for 24 to 48 hours, then take the ram out. And then we would know 140... They were gestation.
Oh, so they do use a ram, isn't it? Not artificially inseminated.
That's the boy right in there. We used a ram. They were not. It's difficult to do AI in sheep.
Is it?
Yeah, and you have to do it laparoscopically.
Right.
And there's no reason. We didn't
There's no
need
reason
to.
to.
There's no reason to.
No.
If your ram is healthy, you're fine. So we cycled the ewes, exposed them to the ram for 24 to 48 hours, and then their gestation's 147 days. So at day like 143 or 44, we would give them a shot, and 18 hours later do a cesarean.
All right.
So it's called cesarean derived. It's done in rabbits and dogs and mice and rats and some things like that, smaller things. But it hadn't been done in sheep,
Right.
because, or if it's been done in sheep, they never brought the offspring to adulthood.
Right.
Okay, they might do a cesarean derivation and do a vaccine study or a medicine study, and then when they were, 8 weeks or old or 16 weeks old, they'd just mix them in with the other sheep because they have to become ruminants. Right, in order to digest the cellulose. They're not ruminants until their rumen has been seeded by the microbes.
Yes.
In order, we don't digest grass unless we have these microbes.
Yes.
So in the case of the sheep, they're ruminants, and they have these microbes that they get from other sheep, and all they have to do is be sort of in the vicinity. They don't even have to be eating out of the same pan or anything, but nose-to-nose contact, they can shed their...
Really? Yeah.
They belch and they chew. They eructate and masticate. That's what they graze all day, and then most of the time, they spend, I think, 10 hours a day chewing their cud.
Yeah.
And all that's doing is breaking down the grass or the corn or whatever is they're eating into smaller and smaller pieces, increase the surface area so the microbes can get at it and break it down and make the byproducts that becomes what the animal actually gets transferred through its bloodstream. So so we did the cesarean derivation, hand raised all these babies, found a way to transfer the microbes without transferring disease, which would be like the IT part of what we've done here. And then once we had it established, then they can just share their bugs with whoever else comes along. So
Is that a trade secret?
that would be a trade secret. Okay. Yeah, we did we had to do something special and handle the stuff in a special way in order to do that without transferring disease. So that's why people do not grow ruminants to adulthood that are this high level of specific pathogen free, because at some point they're going to have their microbes.
So what sort of pathogens are we talking?
They are free of so many things. They've never had a cold. There's never been a runny nose in any of these sheep,
Really?
which is incredible. In fact, as a veterinarian, I swear, I thought a runny nose was normal in a sheep. That's how often you see it. And their noses are dry. I mean,
Wow.
they sort of need their noses wiped a little bit. They've got like feed on them and stuff like that, but normally you would see the nose has just got this runny stuff constantly.
Really?
And these don't have that. So no pulmonary disease.
Where do I sign up? I could use that.
I know, really. You had to be raised separate from other people.
It's too late, David.
Sometimes I wouldn't mind that.
You know how I compare this? I say it's like when your kids go to nursery school, like people's sheep flocks, they're not like riddled with disease and things like that. I mean, they're just normal. Like your kids catch a cold, the sheep catches a cold. It's not like they're alarmingly ill. conventional sheep are just conventional
Yeah,
sheep.
I mean, there's not a.
And so I compare it to when kids go to nursery school in the fall, they all trade back, the bacteria and viruses, and they, the runny nose happens, and then they get over it, and that's fine. But when you have sheep that are being mixed together for a research project, and then they have, then they're trying to measure something or do a procedure on them, and they get a cold, suddenly the data is compromised, or the animal gets sick, and so that doesn't make for good use of resources at all. And that was, they were having trouble getting normal baselines in the pulmonary sheep in particular, the pulmonary studies. And so, and I'll tell you, when I saw that blood collection study, and they showed the beginning parameter point, all the sheep were like in one point. It was beautiful. There was none of the scattering and standard deviations of this and that. I mean, they were all the same, and it was wonderful. I was like, wow. And previously, they had the scatter of baseline points, and it, who knows what's going to happen when you have that.
Once,
So.
once these are delivered to the end user, how do they have to take certain precautions to keep them free of disease as well?
Well, we've told them that if they have any other sheep, and I mean, these they're not like super sheep as far as their immune system. Their immune system is like any other sheep's immune system, we think. No one's really studied it.
But doesn't an immune system have to be primed by infection? You mean it's ready to be primed if it...
Right. If it gets, well, see, now they are exposed to environmental things here. Okay.
Yeah.
What we've stopped the transmission of are the sheep-to-sheep pathogens. Okay.
Right.
So they can get an E. coli diarrhea, for instance.
Okay.
That could be, because that's in the barn, and they can get things from the soil.
Yeah.
So so they have an immune system that has work to do.
Okay.
But when but we tell these people, when you bring your animal into your institution, you want to do a real big cleaning and disinfection before you bring them in if something else has been in there. And they can do gas sterilization of rooms and all this kind of great stuff, when you've got a nice concrete square room. And then, but be cautious of what you bring in. If you bring in other sheep, if they're not of the same health status, then you could transmit disease, and there's no reason why they wouldn't get it. But what we've done here is this is a barrier facility. We've maintained a barrier between our sheep and other ruminants. Has been the main focus. But really, I mean, we worry about anything that comes in this farm. We don't have a cat for rodent control, for instance.
All right.
Because I don't want a cat in here that could give them toxoplasmosis.
Mm-hmm.
So, there are some diseases that are transmitted from deer or dogs or cats or something like that.
No, dogs are wonderful.
That sheep could get.
Are there rodent-transmitted diseases?
Well, things like salmonella, sure.
Are you able to keep rodents out?
So we do rodent control. The other piece of what we do here
Keep
is
it clean,
surveillance.
basically what we do.
And we do a lot of surveillance. So even though we've done this cesarean derivation, how do we? We have to continue to do the surveillance to demonstrate our biosecurity is intact. Because, like
We've
you
never
said,
had a
we
problem.
bring hay in.
We'll still do rodent droppings every once in a while, but
Yeah, we do a lot of cleaning.
keep things pretty clean.
If you spill the grain on the ground, you got to sweep it up, basically. Yeah, it's a different.
Is there any research in xenotransplantation?
Oh, well,
with these animals?
mostly
He worked with the transplant group at MGH.
Massachusetts. We have one place in New York.
They did
Orange
swine
Grove, New
and
York,
and
which is just over the
baboons
Tappan
at
Zee Bridge.
MGH, but there is a
So
big center at
those animals
MGH
we
that
all
does
deliver
transplant
all the
studies.
animals ourselves.
But not with sheep or
And
not?
then
But
we
I
have
don't
one
know.
place out in
I was
Arizona
gonna
that
ask
we
him.
fly them out to. So we what's that?
Sheep xenotransplantation, are they doing that in anything?
There's a guy out in Texas, and Johnny.
So Dick's background is laboratory animal medicine.
That has-
My background is veterinary preventive medicine. So he's worked in lab situations. You know, takes care of the research animals. That's his specialty. So you were saying about somebody in California?
It's Texas.
Texas.
He has created chimeric organs in sheep that are part human, part sheep.
Wow.
What organs do you know?
Liver,
Liver.
primarily.
What does that mean, part human?
What he would do he would inject actual human cells into the fetus of the lamb that would populate the organs in utero. So the lamb would be born with being tolerant to the human cells.
Wow.
So that the liver wouldn't be as antigenic to a human as a
Pure sheep.
pure sheep. But most of the xeno are in pigs. They have herds of pigs that have had antigens removed from their surface of their cells by genetic manipulation that makes them less antigenic.
But is that basically just a sort of historic accident that it happens to be pigs, or are pigs?
Well, again, pigs are more commonly used in lab in research, and I think a lot of that is they're much easier to raise, to breed.
Well, their gestation period's 3 months.
3 months instead of 5.
And they have litters. They'll have 6 or 8
Oh, yeah.
eggs. So there's a difference.
Yeah,
right
the one I
there.
saw giving birth had 12.
That's right.
Yeah, so they
10,
they,
12,
they're
14.
much more prolific. So they, especially the mini pigs, and their size is comparable to people. But other than that, I mean, I think that was just.
So there's no intrinsic genetic reason why
Well, I mean,
pigs
they're
are better.
monogastrics.
Oh, right.
Yeah, I'm not sure if these guys are more antigenic. If it's easier for a human to accept, or they haven't done anything. They do most of their xenografts into primates, but I don't know if these guys are more difficult because they might be further separated and
Genetically.
Genetically,
I
right.
thought that there was some attempt to use pig hearts for heart transplants or something. I mean, the pigs were used.
Yeah,
for-
It's actually been done, I think,
yeah,
isn't it?
yeah,
Yes.
yeah.
Not with any great success.
Not
is
a,
it?
I don't know if they've done it in people yet. They did the baboon heart into a person. We've done some pig transplants into baboons, into primates in research. Mostly when they do that type of thing, though, it's they'll put the pig heart in the abdomen, so they'll just hook it up to the aorta, so it's not life sustaining.
Right.
So then they could study the immunology,
Oh, I
the
see.
injection process,
Yeah.
and how the muscle will function. But I mean, you get animals you could
Palpate the heartbeat
get a
on the
heartbeat.
abdomen.
Yeah.
Yeah.
I can feel his pulse.
Oh, yeah.
You
They
know
have
what?
little.
It's getting cold. Let's just do a little look here, and then we can go in the office where
Okay.
it's warm. Because I see you going, ooh.
I was just so fascinated.
So we added this facility like in 2010-ish.
Oh, wow.
We tripled our animal housing. I mean, that's just 7 little pens out there. And so that barn morphed into our lambing barn. So ladies in waiting, ladies with babies, and then weaned babies are in there, so we can keep a close walk on them. They're just outside my office, right? And then here's another breeding pen, and she's really clever, Red. Okay.